DNA mutations. So what is a mutation? A mutation is any change in an organism's DNA. Some mutations are going to affect a single gene, and other ones are going to affect an entire chromosome. What can cause mutations? There's a bunch of different stuff. It could happen during replication or cell division. It could happen spontaneously, so sometimes they just kind of get a little error in the middle for no reason, or it could happen because of a mutagen in the environment that you're exposed to, like UV radiation or a chemical. Some mutagens are going to affect the entire chromosome, and those are going to be the ones that tend to happen during meiosis. Replication errors happen if DNA polymerase does not fix a small number of errors. They build up over time, and eventually they will affect how the cell works, and if each cell in the body is doing that, the entire organism will start to function differently. For example, a bunch of different studies suggest that mutations are the main cause behind why people age. Mutagens are agents in the environment that can change DNA. They speed up the rate of replication errors, in some cases even breaking the DNA strands apart or breaking them down completely. UV rays and sunshine and industrial chemicals are going to be the two most common ones that we run into. Some kids are exposed to radiation and different things. So the Chernobyl disaster that happened a few years ago, there were a bunch of kids who wound up getting this weird ability where metal could stick to them, sort of like they were magnetic. So I wouldn't say it was beneficial so much as it is just kind of weird or extraordinary that happened. Mutations in the gene that control the development of the rod in your eyes gives you night vision, so some people are actually able to see in the dark. So just because you have a mutation doesn't necessarily mean it's going to be anything bad. You could wind up getting night vision, which is kind of cool, or you could be like Magneto. Alright, two major types of mutations. So you've got gene mutations, there are two main types there, and you have chromosomal mutations, there are 4 types there. The two types of gene mutations, you have a point mutation and you have a frame shift mutation. A point mutation is going to involve a change in only one base, and a frame shift mutation is going to mess up everything that happens after that mutation because there's an insertion or a deletion. With a point mutation, there's going to be one substitution, so one base gets changed to another base. So it would be like incorrectly writing a single letter in a sentence. So you have the ape saw the dog beg. Makes sense, totally normal, not a problem. Down here in the second one, if you were to copy it over and you made a mistake, it would be the ape saw the doe beg. So the letter G got kind of switched up during the replication process and was replaced with the letter E. It changes the meaning of the sentence, but the only part of it that actually changed was this word right here. So if you're going through and noticing that each one of these words is only 3 letters long, that would mean that they're basically like a codon, so this one particular codon is not going to work correctly. Now, you know from your activity that if you have a codon, it's going to code for a specific amino acid. If that letter is changed, you have a chance of getting a wrong amino acid, which means that the protein that you're making will not function properly, if it's able to function at all. So in your notes, you need to circle the substitution. So what's going to happen? A substitution is either going to cause a single amino acid change or no change at all. Remember that many different amino acids, or different codons, I should say, will code for the same amino acid, or they can even code for different ones. So if GAU changes to GAC due to a mutation, the resulting amino acid is still going to be aspartic acid. It's not going to change. So the effects of a point mutation, so if you notice right here, on the right-hand side, you have one little point mutation, and you'll notice that caused glucine here to turn into asparagine there. So it's not a gigantic difference, it's not like it messes up the entire protein, but even changing just one amino acid will change how a protein functions. Frame shift mutations are a much bigger deal. You'll have an insertion, or you'll have a deletion. With an insertion, there's one more base that's added to a DNA sequence, so it messes up everything down the line from there. So you have the ape saw the dog beg, but what if we went through on accident and inserted a letter E right here between the A and the W? Then it becomes the ape say with a so it's not even a sentence anymore. The entire thing gets frame shifted over because this E was inserted, so everything that came after the E gets sort of jacked up a little bit. It all gets slid over just one spot over. You wind up with these weird amino acids, if that was going to be a codon sequence, that don't make any sense. A deletion is the exact opposite. So instead of adding an extra base, you're taking one out. So one or more bases are removed from your DNA sequence. So instead of the ape saw the dog beg, it would be the ape saw, but the W gets removed. So everything else has to shift over one spot to the left, because remember, your frames, You're reading codons in groups of 3. Everything moves over a spot to the left, so now it's the ape sat head, ogb, egg. It doesn't make any sense again, alright? So everything gets shifted over a spot to the left with deletion, and with insertion, your reading frame shifts over to the right because you have an extra codon. So what's going to happen if you have a frame shift mutation? Well, everything after that base that gets moved is going to be mutated. It will affect every single amino acid after that one mutation. So if we go through and notice that there is a deleted base here, then everything, all of this on both sides is going to be messed up. So instead of getting leucine and then all these other amino acids in a row right here, you'll notice that these last two are not the same as what they're supposed to be. It will wind up affecting the entire protein. Most of the time that this happens, the protein is not functional, or it will be functioning in a completely weird way, and it can really detrimentally affect the organism in which it occurs. So when the sequence of DNA bases changes, the resulting mRNA from the transcription is going to be different. If the mRNA is different, the amino acid sequence is going to be affected. So all sorts of horrible stuff can happen as a result. So again, you can have a substitution. So instead of having a T, they put in an A, and you'll notice that methionine stayed the same at the beginning, but then arginine and histine were swapped out. So just by switching that one letter, not such a big deal. The only thing that changed really was the arginine versus histine. And an insertion, so you randomly throw an extra letter in here, and it messes up the entire amino acid sequence. If it was supposed to look like this, now everything gets shifted over to the right, and so you have a completely different set of amino acids. And with a deletion, you go through and remove a letter out of the entire sequence, and so as a result, nothing makes sense after that point. The words aren't the same anymore, or the codons aren't the same anymore. With chromosomal mutations, they're going to involve a change in the number or the structure of the chromosomes. You have deletion, which is where all or part of a chromosome is just completely removed, lost. You have duplication, which is where they go through and accidentally repeat themselves in a chromosome. It can happen one or more times in the same chromosome. You have inversion, which is where they pull off part of the chromosome and flip it around and stick it back in the wrong direction. And you have translocation, which is where one chromosome breaks off and attaches to another non-homologous chromosome. So when we talked about crossing over, it would be like if crossing over occurred in two different chromosomes that were not supposed to be near each other. So they sort of swapped information with the wrong partner. So here are a couple examples of what your chromosomal mutations would look like. If I've got deletion, you'll notice that B is now gone, so there's no B here. If I have my duplication, I'll be adding an extra B in here, so now I've got BB. So it just kind of messes up everything else that happens down the line in that chromosome. If I look at inversion, you can see that the ending piece got completely flipped around. So instead of being A, B, C, D, E, F, it's now A, E, D, C, B, F, because these two sort of swapped their location. And then with translocation, they have completely missed the mark here. They grabbed the wrong partner when they were doing crossing over, and as a result, you wind up with a sequence of bases that don't make any sense. It's supposed to be in alphabetical order, and it skipped over a bunch of stuff on that one particular chromosome, and now as a result, all of its functions are going to be negatively impacted. Now, mutations can happen in body cells. They can also happen in your sex cells. So the big problem is that if it happens in a body cell, does it really matter that much? Yes and no. It's bad for you as an individual, but just because I have a mutation that happens in the cells on my arm doesn't necessarily mean that that's going to affect how my offspring are able to grow over time. It's not going to be something that I pass down. So like if I go tanning a whole lot and UV radiation causes me to get melanoma, I'm not going to have a kid that has melanoma just because I have melanoma as a result of being exposed to UV exposure. It's not going to work like that. My gametes themselves, so the sperm or the eggs of the individual, have to be negatively impacted in order for a mutation to be passed on to the young. Mutations in gametes or sex cells can be harmful, or they can even be beneficial to the offspring. These mutations are going to be the underlying source of genetic variation, which is what we're going to be talking on in our next unit. Genetic variation is a fancy way to say adaptation. A bunch of adaptations over time is basically evolution. Often the effects of mutations in a gamete are so harmful that the offspring doesn't develop properly or dies before they can even reproduce. So if you wind up having weird mutations, and I'm sure you've seen pictures before of like a calf, like a baby cow, that has extra limbs or an extra head, all sorts of weird stuff, Those are considered mutations, and as a result, what winds up happening is that organism isn't able to fend for itself, it's the weakest one in the herd, it gets picked off by predators, or it can't reach its food source, it starves to death. All sorts of horrible things can happen as a result of mutations. So natural selection is going to be the one that's going to remove that mutant characteristic from a population. They're not able to adapt to their environment, they become a large target for predators. More rarely, a mutation can result in a more beneficial characteristic, and these mutations are favored by natural selection, and therefore get passed on. So if I develop some sort of weird mutation in my gametes, so when my offspring are born, that allows them to, I don't know, eat less food but get more energy as a result, that's something that would be advantageous. So their offspring are going to hopefully acquire that same trait, which means that you would have a whole group of people or a whole group of organisms that could eat less food but get more work done, and that's something that would be beneficial. Just an example that I'm coming up with off the top of my head. There's a bunch of other ones that actually occur. Repairing DNA can occur. So you know that you have enzymes like DNA polymerase that go through and proofread the DNA, and when it proofreads it should replace all the incorrect nucleotides with the correct ones. These repair mechanisms work extremely well, but they're not perfect. They do not work 100% of the time. The greater the exposure to a mutagen, such as UV radiation or UV light, the more likely the chances that a mistake is not going to be corrected. So DNA polymerase is great at fixing a couple of errors here and there. It can catch them. It's not going to catch every single one of them. So you want to make sure that your original sentence, like when we had before with the dog saw the cat bag, you want to make sure that that sentence stays as close to normal as possible so that you can catch it and be like, oh, this is what it was supposed to say. If it's all jumbled, it doesn't make any sense, DNA polymerase won't even know how to fix it. So that's it for your notes. Thank you for watching, and I'll talk to you later.